pretty boring because you've probably heard all of it before. We're talking about diffusion, membrane transport, osmosis is part of membrane transport. Maybe you haven't really studied too well facilitated diffusion or primary or secondary active transport. Those might be pretty new to you. My task today and tomorrow is to not just remind you of these types of membrane transport, but also to spin a clinical perspective. Why do you care to hear about diffusion again and osmosis again? Well, now let's hear about it again, but from a clinical perspective. A lot of disease states exist because there is a flaw in membrane transport. Diabetes mellitus comes down to a membrane transport problem. Diabetes mellitus is a very costly disease in this country. We are the second most obese country in the world. We're number 2. Mexico, and they beat us out about 3 years ago.
Wait, with weight or diabetes?
With weight, obesity.
Really?
We used to be number 1. We are not number 1. I don't know if we've reclaimed number 1. Arguably, this is a point where we should not want to. We're number 1! We don't want to do that. Every year, there are hundreds of thousands of new diagnoses of diabetes. So not too long ago, we were at 20 million plus and growing for diabetes mellitus. Most of those are type 2. And you will learn all about type 1 and type 2 diabetes in this unit. Type 2 diabetes, arguably, by the time I'm finished teaching you about them, you might agree with me. I'm saying arguably, type 2 diabetes is preventable and can be reversed. Arguably. Type 1 diabetes is pancreatic failure. We no longer call them insulin-dependent or insulin-independent diabetes mellitus. That's an old way of saying it. We don't say juvenile onset or adult onset anymore, because we can have adults develop pancreatic issues later in life. They don't have to be kids. And we have, because we're the second most obese nation in the world, more and more of our children are getting type 2 diabetes, which was considered to be an adult issue from overeating and being lazy adults. We're number 2. We've got a lot of obese children. For whatever reason, we can point fingers at so many things, but more and more of our children are becoming obese, and that's a very sad thing. So I'm not going to go through this whole lecture, even though it is a very short lecture. I'm going to keep you on the same timeline as the early afternoon lecture. It's probably easier for me to ask you, what doesn't the cell membrane do? What doesn't it do? Well, it doesn't contain DNA, it does not participate in gene expression, but it does house products of gene expression. You can see proteins embedded in the cell membrane. And we're going to talk about the different kinds of proteins that you can find there and what they do. I'm going to go through all the components of the cell membrane, and again, arguably, probably the 6th time you've heard about this in your lifetime. I'm going to tell you about phospholipids and what they look like chemically. I'm going to tell you about these proteins. I'm going to tell you about the sugars that you see attached to the proteins and the phosphate heads. I'm going to go through the anatomy of the cell membrane, which is really boring. However, in order for me to teach you why you care from a clinical perspective, you need to remember that anatomy. It's really important. What you see in the cell membrane provides the following functions. The proteins can act as receptors. We know that from Unit 1. The proteins can act as enzymes. They can facilitate reactions. The proteins can actually move substances from one side to the other. They can act as carrier proteins or channels, as you learned today. The proteins that you see there can also act as adhesion molecules, keeping two cells stuck together. That's really important when it comes to cardiocytes. Cardiac tissue has a very special kind of contact between two adjacent cells called intercalated discs. I know you've heard of that before, but it comes from proteins aligning just right and creating pores from one cell cytoplasm to another. This allows the electrical current that I taught you today to travel from one cell to the next directly. And that allows your heart to beat in what's called syncytium, which is a very big fancy word. It means togetherness, coordinated. The proteins can also be used, and the sugars can be used as recognition molecules, self versus non-self, just to name a few things that we've already talked about. So let's start with a phospholipid. A phospholipid is considered to be amphipathic or amphiphilic. Let's break it apart. Amphi, like amphibian. What do you think of if I say amphibian? You might give me a specific example, but what does amphibian actually mean to us? It's an animal that loves both water and land. It's amphibious, it likes both. So an amphipathic, amphiphilic, loving both water and lipid environments. The phosphate heads that you are seeing here in this bigger picture and drawn here as the blue circles, they are water loving. When we talk about the phospholipid bilayer, we know that we have two layers of lipids, phospholipids, that orient their heads towards body fluid compartments. You see, that's a new way of looking at it, isn't it? You now know your body fluid compartments. One layer of the phospholipids faces intracellular fluid, and the other layer faces extracellular fluid, which could be interstitial fluid in this picture. So that's a new level to your learning. Attached to the phosphate heads, we can have other water-loving groups. We can have nitrogenous groups, we can have proteins attached, we can have sugars attached, and I'll talk about that in just a moment. The fatty acid tails, these long hydrocarbon tails, they face each other. And I like to think of the phospholipid bilayer as a well-made peanut butter and jelly sandwich with the two bread pieces facing the external environments, water environments, I should say, in my example, extracellular fluid and intracellular fluid. And the gooey deliciousness middle layer, the peanut butter and the jelly, think of that as the phospholipid, as the fatty acid tails of the phospholipids. That creates a zone of hydrophobicity, meaning water hating. Those tails don't want to face your body fluid compartments. They face towards each other. And that means moving through the cell membrane, if something's going to move from water through water-loving heads, through water-hating tails, through water-loving heads again into a water fluid compartment, it has to be able to move through the fatty acid tails. And in chemistry, you might have heard like dissolves like. Something that is hydrophobic, water-hating, can move through this thick layer very easily. But something that is water-loving is going to have a hard time getting through this unless it has the right chemistry. And we're going to talk about that tonight. When I get to that chemistry, that's when I'm going to stop, and you're going to weigh your bags. So here's our phospholipid bilayer. And again, we have proteins. If proteins go from one end to the other, if they span the entire distance, this is called an integral protein. If a protein is found associated with one side or the other, it's called a peripheral protein. And in this picture, you're seeing peripheral protein that can change conformation, change shape, and even flip to the other side. And that kind of protein changing its shape is acting as a carrier protein, delivering a signal it has bound to the inside of the cell in this example. We're going to talk about how these signals can be carried to the inside of the cell. Here is another example of a peripheral protein that is found within the cell in this example. Here I'm showing you that potassium is found abundantly, so this side is the inside. And those internal proteins can act as enzymes and also participate in this relaying of the signal from the outside to the inside. Again, that is more for us to learn in Unit 2. Associated with the phosphate heads on the external side are sugars. They can be attached to the proteins or the phospholipid heads. And these sugars can also be associated with proteins that are just kind of freely floating just outside of the cell. And this sugary area just outside of the cell is called the glycocalyx. Now, these sugars that are just outside the cell, they are negatively charged. Please understand, the inside of the cell, as you learned tonight, is still more negatively charged than the outside. So yes, these are negative, but the inside is still a lot more negative than that glycocalyx. From a perspective of inside versus outside, we would say inside being more negative, outside is relatively then more positive. It's a lot like talking about your debt. Would you rather be minus $50 or minus $5? Both are negative, agreed? But one is less negative than the other, closer to positive. Are you starting to get my analogy? Working way too hard tonight. It's just a sign of the week to come if Monday I'm working this hard. Embedded in the phospholipid bilayer is cholesterol, and these lightning bolts that are shown here in blue are to represent cholesterol. Cholesterol gives structure to the cell membrane, which is weird when I say that although it gives structure, the cell membrane is still fluid. Thank you. These proteins that are here, they're not fixed in place. They can move through the cell membrane. It's dynamic and fluid. It's like icebergs in our ocean. They're not anchored in a particular place. They move. We know that tragically. History has taught us that. Not if you're still with me. Okay, good. I say iceberg, you say good. Okay, so you are with me. Okay, good. I was a little worried. So the cholesterol is like antifreeze. It helps to maintain the cell membrane in a fluid state, but at the same time, it does give structural support. So some sort of rigidity. And that sounds like it's an oxymoron, but that's what it does. Flexibility and stability. The cholesterol is a kind of lipid, and I'll remind you that cholesterol is a precursor for your steroid hormones that you're being tested on Wednesday. Now, you're doing a lab on membranes, and you are exploring a separation of solutes across a selectively permeable membrane. You heard me say that those pores that are found in the dialysis membrane are too small to let the sucrose move. So your membrane that is in front of you right now that you're playing with is a lot like this picture. What can move through your dialysis membrane? It has to have a certain molecular weight. It has to be small enough to get through the pores. Okay? There are very small molecules that can get through our cell membrane, but it's not just about size. It's also about their chemistry. They're not polar. They can get through that hydrophobic core. We can have small molecules that are somewhat polar. Water is somewhat polar. Water, being water, is water-loving, How does it get through the hydrophobic core that I just told you about? It's small enough and not charged radically. It can worm its way through the cell membrane, which is why I've been saying to you, if we have an equal particle count per unit of volume, if the particles cannot move, what will? Water, because water can worm its way through. Tonight you're playing with sucrose. This is a very large molecule. It is water-loving. It's not going to get through your cell membranes that are in front of you tonight very well because it doesn't have permeability. But then what about electrolytes that you learned about tonight? Those are very, very small, but they're charged, very charged. They don't get through very well. But then you're all looking at me like, Kara, but we talked about electrolytes getting through the cell membrane and having permeability. Yeah, the membrane in front of you right now is different than a biological membrane. How is it different? How are ions and even sugar getting through your cell membranes in addition to these gases? What is it that a living cell membrane has that those membranes in front of you do not? Go ahead.
The voltage gated canal?
Okay, so you're giving me a very specific example. In general, what are those things that are going through the cell membrane that I said could be integral or peripheral? Proteins. So a living cell membrane is different than what's in front of you in that it has gene expression. It can insert pores that are proteins, and those proteins can be gated, as you learned tonight. They can open, they can close, and if they open even for just a moment, we can have those tiny but charged electrolytes flowing. And if we have ions flowing, what's the scary word that you learned that described that? A flow of charge. It's a current. And if the gates are closed, that means resistance. If your light is red, you are met with resistance. Your gate is closed. If your gate turns green, that means you're allowed to now conduct through the intersection. You are flowing. And if you are a charged particle, that's electricity. Electrical events precede mechanical. Many of you using your phones, your computers, they cannot do work for you. That's the mechanical event without them being charged first. That's the electrical. So please measure your backs.